Theiler's virus causes a biphagic disease in mice. 1 week after intracerebral injection of virus, Theiler's virus infects neurons and causes acute polioencephalomyelitis. This is immunohistochemistry against the virus. You can see virus antigens not only in cell bodies, but also in processes of neurons. The virus spreads in the central nervous system using axonal transport. And 1 month later, the mice develop a chronic progressive demyelinating disease and show paralysis. During this chronic phase, the virus persists in the spinal cord. This is how we process the spinal cord of mice. Spinal cord was cut transversely. If you see the sections of the spinal cord, butterfly-shaped gray matter exists inside, and it is rich in cell body of neurons. The white matter locates outside, and here is rich in myelin and axons. In this luxofast blue staining, myelin sheaths are stained blue. This is a time course study of myelin staining of the spinal cord in Theiler's virus infection. Obvious demyelination is first detected 1 month after infection. These white vacuolar areas are demyelination. You can also see inflammation around vessels in the white matter. In contrast, during the first 3 weeks after infection, most areas of the white matter appear normal, and only mild cell infiltration can be detectable in some areas. However, in the next few slides, I will show you many damaged axons in this normal appearing white matter. We detected axonal damage using immunohistochemistry against neurofilament. Neurofilament are phosphorylated in the cell body of neurons and are transported to axons. So in normal neurons, the nonphosphorylated form is detected in the cell body, and the phosphorylated form is detected in axons. When neurons and axons are damaged, you can see the opposite distribution of phosphorylation. The phosphorylated neurofilament in the cell body and the non-phosphorylated form in axons. In the next slide, I will show you this non-phosphorylated neurofilament staining. 1 week after infection, most areas of the white matter showed no staining. However, you can see axonal swelling positive for non-phosphorylated neurofilament in the lateral funiculus and the ventral root exit zone. In higher magnification, you can see retraction boards, an indicator of axonal damage. The number of damaged axons increased over time. You can see beaded and fragmented axons. As I showed you 2 slides ago, this area of spinal cord appeared normal by conventional myelin staining. Since obvious demyelination was detectable 1 month after infection, these results suggest that axonal damage precedes demyelination in Theiler's virus infection. Next, we examined whether damaged axons co-localize with demyelination at a single fiber level using confocal microscopy. We visualized damaged axons shown in green with myelin sheaths positive for myelin basic protein MVP, shown in red, 2 weeks after infection. A merged image shows distended damaged axons wrapped with myelin sheaths in the white matter. This shows that axonal damage precedes demyelination. In this slide, you can see a virus-infected oligodendrocyte in red and normal axons in green, stained by phosphorylated neurofilament. Here, one single oligodendrocyte wraps multiple normal axons. You can see red myelin sheaths outside and green axons inside. Interestingly, the top myelin sheath, pointed by a large arrow, lacks normal axon stain and looks empty. Let me explain again. This is one single oligodendrocyte that wraps several axons. The lower four myelin sheaths contain axons, but the top one does not contain the axon. This suggests that the axon is either damaged or lost before myelin destruction. Therefore, this is another supportive evidence that axonal loss or damage precedes demyelination. In the next slide, I will show you this empty myelin profile in multiple sclerosis. I borrowed these slides from Dr. John Rhodes, Department of Neurology, University of Utah. In this slide, axons are visualized in red and myelin sheaths are stained green. If you look at the left figure, from the normal appearing white matter of an MS case, you can see red axons wrapped with green myelin sheaths. In contrast, on your right, from some MS patients, you can see myelin sheaths that lack axons. This is so-called empty myelin profile. This suggests that in some MS cases, the lesion can develop from the inside axons to the outside myelin sheaths. This figure is from another case report of an MS patient, published in Neurology. Myelin sheaths are shown in green, and axons are shown in red. These normal myelin sheaths contain axons, but some other myelin sheaths contain no axons. These results also suggest that in human patients with MS, axonal damage can also precede demyelination. In Theiler's virus infection, we also look at how macrophage and microglia in the spinal cord respond to axonal damage. 2 weeks after infection, we can see activation of microglia that have dendritic processes. 3 weeks after infection, microglia and macrophages change their morphology. These microglia and macrophages seem to take up degenerated axons and become rounded phagocytes. The number of reactive microglia macrophages increased over time. Based on the results, I have proposed a new model of lesion development in Theiler's virus infection, the inside-out model. First, axonal damage occurs inside, then demyelination follows. Here, the lesion develops from the inside to the outside. I just proposed that there are two hypotheses how axons are damaged in MS. The outside-in model for an autoimmune disease, EAE, and the inside-out model for Theiler's virus infection. However, more recently, there are experimental reports showing that some form of EAE can develop from inside out. In this new model for EAE, mice were immunized with neurofilament light chain protein in complete fluorescence adjuvant. The mice develop autoimmune responses against axons and show spastic paralysis. If you look at histology, you can see axonal loss and swelling with demyelination. These results demonstrated that if you induce autoimmune responses against axons, the lesion can develop from the inside axons to the outside myelin. I would like to emphasize that these two hypotheses are not mutually exclusive. For example, in conventional EAE, first lesions develop from the outside. Anti-myelin autoimmune responses initially attack myelin, and axons are damaged secondarily by severe inflammation. Then, if axons are further damaged and severed, the distal stump of axons will degenerate. This is Wallerian degeneration. From here, the myelin sheath wrapping degenerated axons will also degenerate. At this stage, the lesion develops from the inside axon to the outside myelin. Then this degenerated myelin will be taken up by microglia and macrophages, and myelin antigen can be presented to autoimmune cells in the brain. And the activated autoimmune cells can attack myelin from the outside. This will lead to the second cascade reaction and disease progression. If this is true, this vicious cycle can occur regardless of whether lesions develop initially from the inside or the outside. It is also reasonable that two types of lesions can be seen in one MS patient. These slides are again borrowed from Dr. John Rhodes, University of Utah. In MS, you can see two types of lesions. On your left, this is a typical demyelination in MS. You can see naked red axons with no myelin sheaths, which supports the outside in model. On your right, you can see green myelin sheaths contain no axons, which supports the inside-out model. But if you can carefully see the slide on your right, you can see naked axons. So two different types of lesions can be observed in the same or adjacent area in MS.